My name is Stuart Early. I'm based at Northern Kentucky University. I also work with Waters Corporation, and I'm a analytical chemist by training. So I've done a lot with cyanobacterial toxins. This we're going to talk about is looking at it from recreational waters. It's a very big, challenging area, recreational and intake water. This was some work I've done with Wendy Strangman and Katerina D'Souza at UNC Wilmington as well. So great background from Jordan before on the background of the algae toxins, so I don't even have to cover that. I'm just going to go into the analytical detection method we use, which is mass spectrometry. Typically, toxins can be analyzed using ELISA or some other more general techniques, which are very good for screening. The one challenge is you don't get the exact species of microcysteines or cylindrospermopsin or other type compounds and toxins that are produced. You just get a present or not present in an approximate amount, but microcystine LR, LA, and all the different structures that are associated with these all have different LD50s and can have different interactions with toxicity in that. And the challenge as well in recreational waters, like she mentioned, had some great photos there, is, you know, a lot of the stuff that drives that is things such as phosphate, nitrates, and other fertilizers and other things that can feed the bloom. Some people treat it with copper sulfate. We've had a lake that they did that with. The issue is you then kill the toxins, or kill the algae, which then release the toxins. So it's kind of a challenging area. But for the analysis, HPLC had been used, which is a traditional high performance liquid chromatography. We went to a small particle system that used smaller particles. The neat thing about this is you can get a very fast analysis. So real quick, what are tandem quads, which is our mass spectrometer we use? Basically, you have two mass analyzers. Let me do my laser pointer. There we go. So you have two quads under vacuum here, quad 1, quad 2, and a collision cell. The ions are separated by liquid chromatography. You have a toxin coming in. It's the first quad, gets fragmented, and you monitor it in the second one. So these are very sensitive and very selective instruments. So for example, microcystine LR, which is kind of one of the common ones, has a mass typically of 995.6, which is the molecular and protonated ion. So what happens is this mass spectrometer will let only that mass come in at that particular time. It selects only 995 into this collision cell with some argon in it. It fragments and shatters the ions, and you end up with a fragment ion in this case, which is mass 135, which is an ADA adduct or ADA fragment of the microcystine. And then you monitor that. So you end up with a chromatogram or a separation at that particular mass. So what's small particle get you? Typical HPLC runs. The lower this is, the better your selectivity. It's like limbo. So how low can you go? The nice thing too is you can, at this point here, you're have very good separation of these compounds, and you can go very fast. So what's it give you? A separation like this. So this is 12. This is our initial work with 12 of the base microcystines, anatoxins, cylindro. The runtime's less than 9 minutes using this separation. To compare what it looks like by HPLC, HPLC runs would typically be 30 to 35 minutes. So in the time it took for one peak to come off by HPLC, we're able to do this by UPLC. They're much sharper, more sensitivity. I can also, as more toxins became available, I can add that to the list here and start analyzing for more microcystines, aminopeptins, microgenins, anatoxins, cylindro, all sorts of different ones. And we've expanded the list over the years. So continue as more standards get available or we get more information on ions to monitor, we can do that here. And you're seeing here now the list of all the toxins that we monitor for in a single run here. So it's kind of nice. You can actually figure out which of the particular microcystines are present. Cylindrospermopsin, we've also added since then 7-epicylindro and some other ones. And we've done some work where we found that in some lake waters. As well as then anatoxin and also anatoxin A. So this is just an example of a reservoir sample that we analyze for. So what you're looking at here is it's a single run. I get 3 masses here for microcystine LA, microcystine LR. It also had the DASP LR. and had anavidipeptin, which is not a current toxin, but it's of interest due to some of the functionality and behavior of it. So this is one you commonly will find in some algae blooms and not in others. But the reason for the multiple masses here is just simply as a way of confirming. So for example, microcystine LA gives you a mass at fragment ion at 135, 102.9, and 86. So if I see all three of those, I have a high degree of confidence that that's the actual toxin that's present. This is the amounts in micrograms per liter that were found in these samples. It was interesting that this sample, usually microcystine LR is the most, but in this case, LA was, which is actually LA is slightly more toxic. So finding more, we went to look at anabinopeptins. There's only a few anabinopeptins that are out there, anabinopeptin A and anabinopeptin B, if I can say that. But they give a common fragment ion, which is at mass 201, which happens to be the CO arginine peak fragment for it. So what you can do is go in and say, I want to look for fragment ions of this and tell me what the parents are, what the original ion was that produced that. So we have a lake water sample here, which does have anabinopeptin A in it. This is a standard, just confirming the retention time here. And we have a lake sample that also had another parent ion at 201 that was coming off later. Turns out the mass happens to agree with anabinopeptin E and F, which both are isomers. So you can actually monitor for that. While you might not have a standard, you can at least monitor samples to see if it's present or not. So it's kind of one of the cool things you could do with the mass spectrometer. So instead of before where I let just the ion of interest in and fragment it and look at a fragment ion, this time I'm letting everything in, fragmenting and only monitoring for this and then saying, okay, once I saw this, what caused it? What was the original parent or precursor ion, as it's called, that formed this? So you can do that with a lot of compounds. Microcystines typically give that add a fragment at 135. I can do the same thing where I monitor for 135 and then look for all the masses that gave me 135 and see if any of those correlate to other microcystines that you may not have standards for, but have been discovered and verified using other techniques. So now we can go back and monitor these samples looking for anabinopeptin E and F, and you can see in the case here for this Lake Erie sample, this Ohio River sample, these samples from out west, they were not present. So you won't find it all the time, but it's kind of neat to be able to actually go in and look for it. Some additional compounds as well that you can look at here. This was looking at additional cylindrospermopsin analogs. So this is cylindrospermopsin, this is deoxycylindro, and 7-epicylindro. Once again, multiple fragment ions. Some only give 2, some give 3. The other neat thing is you can actually look at the ratio of these because these are standards and they fragment consistently. The ratio of these, if they're the same between your standard and a sample within, say, 20% of each other, that's considered a good way to know that you have confirmatory ions as well. It's typically done a lot in drug analysis. So we're using the same principle here of doing ion ratioing, it's called. Maybe. Uh-oh. My screen locked. That's never good. Oh, there. Hang on. Did I lose everyone?
We still have you. You just need to put it back.
I got it.
There we go.
All right. So what I am going to do is stop videoing because that sometimes does that for me. So I apologize. I will go away. Nope, locked up again. There we go. All right. I'm going to run it this way. So hopefully you can still see this because it seems to be locking up on me. I apologize. So one of the things we did was there were some dog deaths in Wilmington, North Carolina, and it was benthic algae. So what we did was we got a pellet sample. We ran it. There was, you can see here, the cylindrospermopsin was present. There was also a sample of cylindro in the pellet that we sent in the standard as well. We also found antitoxin, which might explain for the dog deaths. They were quite sudden. The dog got in and eaten some of the mat or algae and died within several hours. So it was typically an antitoxin poisoning, probably not Cylindro, because Cylindro doesn't behave that way. And you can see here, we can see it in the pellet sample and also in the antitoxin standard, you can see the match for it. There was also, we went ahead and monitored for homoantitoxin. At the time, we didn't have a standard. We knew the mass of it based upon literature searching, and we saw the presence of it. We were able to actually order some. Some was available from Abraxas, so we got it. You can see here the ions. This is just a calibration curve. And you can see here in the pellet, there was around 15 parts per billion of homoantitoxin. This is the standard that was run. So it was kind of a cool way to figure out what was going on. We were monitoring the masses, but didn't have a standard and was able to get one to do that. So in summary, mass spectrometry offers a sensitive and selective method for detecting these toxins. UPLC offers high resolution. You can do MSMS with this and use it for also other compounds. The instrument's used typically for doing pesticides. I also run PFAS on it in the lab. You just have to convert some stuff over, but it's a pretty rugged little instrument to be able to do that kind of stuff. So anyway, I'd like to acknowledge Waters Corporation, Thomas More Biology Field Station, which is where we got a lot of the samples from the Ohio River at, and also Dr. Marion Cannon and Dr. Joshua Cooper all get me all sorts of samples from all over the place. When they drive around and find interesting stuff, I tell them to bring me the samples and we'll try running it. So anyway, with that, I'd like to say thank you for attending. Sorry for my technical issues. I think it was probably the video streaming, but my last take-home message, if I have time, as Captain Jack Sparrow says, when swimming, drink rum, not the water. So with that, thank you.
We have about 3 minutes for questions. I don't see any that have come in so far. I guess I was wondering if you see any co-contaminants with the algal toxins.
Any what?
Co-contaminants.
Oh yeah, you see all sorts of stuff. The nice thing about the mass spectrometer is it can be very selective. So you're looking only specifically for the ions of interest. So this is what's considered targeted analysis. I can also do scanning the background. I find all sorts of other stuff. So that's where I then go off later and look to see what other things are in there, because there's usually lots of other stuff with it. So, but that's the beauty of this. Most of these have common fragment ions, so I can be pretty selective in what I'm looking at. You also have the column that's doing some nice separation up front as well. But for these samples, pretty much I just take the samples and either we do a freeze thaw the lysum and then either filter and run as is or do a dilution if they're too concentrated.
Great. I think there's quite a few that have come in now. Lee, do you want to?
Yep. Are you able to identify the annual trends and concentrations?
Yeah. I didn't have enough time to show. I had a couple other slides, one of which is we do a lot with the Ohio River. And it's interesting. I can do a scan of mass 135, which is the ADA fragment of microcystines. And if you just look at the pattern, it's kind of scary. We have about 4 years of data in which the ratios of the different toxins, RR, LR, and some of the other ones, they almost overlay each other as far as the ratios between the two. It's kind of scary, which makes you wonder if this algae is kind of producing the same thing and the same kind of content over and over. The other weird thing we had is we had some blooms depending on the species. So we had like a Microcystis aeruginosa. I'm not a biologist, so I'm going to really kill these names. That came from Florida. They were typically had LR. The stuff in Ohio River was similar, had LR. We got some samples from out west, which was a Microcystis floss aqua, I think it was. That had absolutely no microcystine LR or RR, but a ton of LA only, which was interesting because if you'd only been monitoring for LR, you would have never known that these other toxin was there. So it's kind of interesting.
We have time for one quick one.
Okay. Can you speak to some of the challenges associated with identification of such large molecules in terms of molecular weight, as well as the large number of conjugators?
The conjugators you just can't keep up with because there's really no standard. So you have to monitor and then kind of make some real big assumptions on there is some really nice Excel spreadsheet that has all of the different congeners that are listed there, and you can pretty much match to it. So that's not really the issue. It's just getting standards and running it. So it's kind of fun. But it is getting a lot worse because we're getting samples now from all over the place, and they seem to be happening a lot earlier and lasting a lot longer. I mean, we had a bloom sample still positive in late November on the Ohio River.
Wow. Yeah.
Which is cold. We're south of you all. So, I mean,
Yeah.
you know, but they still found, like in the, in some of the soil residue or sediment on the riverbank, when we did some extractions, you still had viable toxins when you did lysings.